Next speaker is also from UW, and funnily enough, Troy and Sophie Margridge, our next speaker, had never met before today. And she'll tell us about interrogating protein variant dysfunction.
Thank you. So I am in Judith Vien's lab at the University of Washington, and we're kind of the black sheep here, and that we're interested in looking at protein variants, but instead of using sequencing, we're really interested in using mass spectrometry. And so you can check out my poster at A10. Whoops. Okay, here we go. So mutations can result in lots of different mechanisms of dysfunction, right? A single amino acid mutation can cause changes in solubility, in post-translational modifications. Oh, sorry, I'm used to walking around. Things like small molecule binding and protein interactions. And so this is really important to consider because for many diseases, the severity, intervention, and outcomes can really differ depending on what the mechanism of dysfunction is. And so for that reason, we need to develop methods that can resolve different mechanisms of dysfunction. And this is where mass spectrometry really comes in. So mass spec really bridges the gap between the capacity of deep mutational scanning and the granularity offered by classical biochemical assays. So we can start off with a library of tens to thousands of variants. These are all then pooled. We can perform biochemical assays, and then directly read out protein properties using mass spec. And so if we're directly reading out properties, we don't actually need this like phenotype-genotype link. And so these are properties like solubility, stability, binding, et cetera. And so there's a number of advantages of using mass spec. These are properties that you wouldn't see using typical genomics. There's a number of mass spec compatible assays that we could imagine adapting for these purposes. We're hoping that we can make a single library and perform a bunch of different assays using the same library. And lastly, we can do this all in a pooled format, so it's a lot higher throughput than classical protein biochemical assays. And so as a pilot project, we selected this protein phosphoglucomutase 1. It is a disease-relevant protein for which we have some data already about the stability and the solubility of a number of disease-causing variants. And so all that data is from purified variants, and that's fairly low throughput. And this serves as a really great benchmark for us to now compare, can we measure solubility and thermostability, but can we do it in a pooled format using mass spec? And so we start off with a very modest library of 13 variants, and these are overexpressed in E. coli. The cells are then lysed, this is all pooled, and then I performed a solubility and a thermostability assay. And so I'm going to show you some data from our solubility assay. So on the y-axis, we have solubility. Pink is or reddish pink is our data for the solubility measured using mass spec. And then in blue is from the purified variants of previously published data. And so what you can see here is that we're able to read out the solubilities for these variants. The data presented with the purified variants by SDS Page is kind of like bimodal. Either they're like really soluble or quite insoluble. And what's nice is with mass spec, we have a little bit better resolution, but so we can see a sort of a better range of solubilities, but we can also resolve the highly insoluble variants from the quite soluble ones, which was really exciting for us to see that agreement. The other assay that I worked on is this thermostability assay. And so on the y-axis is the thermostability. Obviously, higher TM this is a protein melting temperature. A higher TM means it's a more stable variant, and a lower TM means it's a less stable variant. And so in blue is previously reported data, and yellow is our data measured using mass spec. And so for 4 of these variants here, what we report is within about 2 degrees what has been previously reported. And then what's also pretty exciting is that these variants, E337K and Q41R, so those are, we didn't have there was no published data on these variants because they're kind of hard to express. And so it was nice that we were able to measure it using mass spec. And there's a number of like technical advancements that are sort of needed to increase the detection. I've only shown you 6 variants here, but there were 13 to start. So we've been working on that, and if you're interested in the technical side of the mass spec method development, I'm happy to tell you about that at my poster. We're working towards adding other selections, obviously expanding our very modest library size, and peptide barcodes. And so we're hoping that the behavior of these variants in our assays is really reflective of what's going on in disease biology, and that we can apply these assays to lots of other proteins and diseases and further our understanding about the proteome. So I'd love to thank everyone in the VN Lab, the Center for the Multiplex Assessment of Phenotype, and if you want to come see a black sheep poster, I'm poster A10.
I have a pretty naive question, which is, I could imagine that, at some point you run into, like with large libraries, just an inability to deconvolute mutations at different sites that are going from, certain amino acid to another just by mass. I guess, then you can digest and things like that. So how big do you think the libraries can ultimately get? And how will you, sort of get around that by mass spec?
Yeah, so obviously with our fairly modest library size, that's not quite an issue yet. But because there's a lot of shared peptides in the backbone, those end up taking up a lot of the signal in your mass spec. So the solution here is to have cleavable barcodes. So then you could just cleave that off your whole protein and then use that as a readout. And that's what somebody in our lab is working on that right now. Yeah.
Also, I have a pretty naive question. I was just wondering, do you think it could be interesting or useful to integrate in your pipeline maybe more advanced mass spec techniques such as HDX-MS, which would maybe give you structural information on your variants? Or would it be maybe logistically too complicated to do that?
Yeah, so from my understanding, HDX is like fairly low throughput. And I don't know if you'd be able to look at like a whole bunch of variants in the same sample all at once. Like the really nice thing about mass spec is we want to be able to look at lots of variants in the same injection. And I'm not sure if HDX would let you do that.
Right. Okay, thank you.
Great.
Thank you.
So much.